Everyone and welcome back to my YouTube channel. So today's topic for the video is important questions for medicinal chemistry part 3, that is in your B Pharmacy 6th semester 3rd year. We have divided the same in 5 units. Number 1, beta-lactam antibiotics. In the class of beta-lactam there are 4 different other classifications included, that is penicillins, cephalosporins. These are the two older generation, and the new also there is certain new generation drugs that belong to class carbapenem and monobactam. Imipenem is an example of a carbapenem. Aztreonam is an example of monobactam. In cephalosporins we have 5 generations, and out of which the 3 generations have further classification as oral and parenteral. They mainly include cefalexins, cefazolines, cefadroxils, cefurexime, cefixime, cefepime. These are the few important few examples of cephalosporin. In penicillin, we have narrow spectrum, broad spectrum, as well as penicillinase resistant penicillin and few beta lactamase inhibitors. So amoxicillin and clavulanic acid, this combination is most widely used because amoxicillin is a broad spectrum antibiotic and clavulanic acid is a beta lactamase inhibitor. So that enhances the bioavailability and the half-life of a drug. And the name of the medicine is augmentin. It is a amalgamation of amoxicillin and clavulanic acid. In beta-lactam antibiotics, basically you have to study about the mechanism of action, the spectrum, and on what microorganisms does it actually act. In beta-lactam antibiotics, if you go to see, mechanism of action may differ from drug to drug. Like in penicillin, it is mainly inhibiting transpeptidase enzyme. This transpeptidase enzyme is responsible for cross-linking in the cell wall of any bacteria. So transpeptidase is inhibited in penicillins. Cephalosporins also act in a similar way by inhibiting cross-linking. So that's really important to study about beta-lactam antibiotics. Next, we have tetracyclines. In tetracyclines, we have oxytetracycline, chlortetracycline, doxycycline, minocycline. Minocycline is the longest acting tetracycline and it has a dimethyl amino group present, additional dimethyl amino group, and oxytetracycline, chlortetracycline, doxycycline are important. The structures you have to study, like for example in a beta lactam ring, you have to study beta lactam antibiotics, the beta lactam ring, and the positions of the groups. You must remember everything that's related to the acyl amino side chain, the substitution at R1 position. Why is the dimethyl substitution important? Similarly, in the clavulanic acid, you have a oxazolidine ring. You don't have a thiazolidine ring. And you lack the acyl amino side chain and clavulanic acid. And in tetracycline as well, you have to study about the structure. It is very, very easy. It consists of 4 rings that are 6-membered, and these rings are fused together just like this. And there are alternate double bonds and single bonds. Here we have substitution R1. Here we have 2 substitutions R2 and R3. Here we have R4. And then the double bonds, you have to remember there is 3 double bonds in the first ring, no double bond here, and there are 2 double bonds here. So it is a octahydronaphthacene derivative. Octahydro because there are 8 places in which the hydrogens are missing since they are fused ring. So you have to remember that it is a derivative of octahydronaphthacene. And then you have to remember this NCONH2OH. All this is important if you go to see about the structure, because they ask you about the SAR, the structural activity relationship of tetracyclines. So you have to remember. And next we have aminoglycans, that is aminoglycosides. Tetracycline aminoglycosides and macrolides as well as chloramphenicol have a similar mechanism of action. They all are disrupting the protein synthesis, but aminoglycosides and tetracycline disturb the 30S, while macrolides and chloramphenicol disturb the 50S. These are the subunits that make up 70S together. 30S is disrupted by, is blocked, we can say, or that it binds on that unit by tetracyclines and aminoglycans. So there are two sites, peptidyl site and exit site, and acceptor site, I'm sorry. And there is translocation of the amino acids. So suppose this is a amino acid tRNA. These are the three amino acid chain. So there is a translocation of this from P site to A site, and then from again A site to P site. So this translocation can be prevented by chloramphenicol and macrolides, but aminoglycoside and tetracycline prevents the binding of amino acid tRNA to the ribosomal complex. So it prevents that formation of ribosomal. So you have to study the difference, and also importantly remember that aminoglycosides are bactericidal, but tetracyclines, macrolides, chloramphenicol, they are bacteriostatic. Yes, of course, there are few exceptions, like at higher concentration, chloramphenicol may act as a bactericidal agent, but at a lower concentration, it is also bacteriostatic. So remember that aminoglycosides is a bactericidal action. Speaking about the spectrum, macrolides are narrow spectrum antibiotics. So you have to remember the spectrum as well. That's all from unit 1 that you have to study mainly. From unit 2, remember the prodrugs. Like for ampicillin, there are two products. Becampicillin, that is a carboxy prodrug of ampicillin. Hetacillin, that is a amino prodrug of ampicillin. Rolitetracycline, which is a pyrrolidine prodrug of tetracycline. It has a pyrrolidine ring. Also, there are many other prodrugs that you have to remember during, you'll study about the same, like ganciclovir is acyclovir is a prodrug. It is an antiviral drug, and it gives various other prodrugs like valacyclovir, ganciclovir, etc. So you have to remember the other agents that are acting, that are prodrugs. Speaking about the macrolides, macrolides is basically including two classification within it. That is, and aminoglycosides and macrolides have similar names, so just see to it that you be careful. Like if you go to see aminoglycosides and macrolides have two, 14-membered and 15-membered. In 15-membered, the most widely used macrolide, that is azithromycin, is a 14-membered ring, while in 15-membered ring, while in 14-membered we have clarithromycin, erythromycin. Erythrocin is the brand name of the erythromycin, so that is also available as an injectable form. Next we have aminoglycosides. So in aminoglycosides, basically you have examples like streptomycin, kanamycin, gentamicin, neomycin, etc. Now just remember that there's no spelling mistake of mycin and mycin. In fact, there is a difference between mycin and mycin. One is obtained from bacteria and one is obtained from actinomycetes. So if you see gentamicin, it has MICIN, but if you see streptomycin, it has MYCIN. So don't think that it's a spelling mistake. These are two different moieties. These are two different sources of the antibiotics. Like chloramphenicol is obtained from Streptomyces venezuelae. Erythromycin is obtained from Streptomyces erythraeus. So just remember the names of the drugs and also the sources of these drugs. That is also very vital. Prodrugs are basically something that is inactive and then it will become active. That is prodrugs. On biotransformation, that is active metabolism. Next we have unit number 3, in which we have Anti-UTI infective agents. So UTI mainly fluoroquinolones and nitrofurantoins. These are the two most widely used classes of drugs in UTI infection. Fluoroquinolones, basically you have nalidixic acid as a quinolone. It lacks fluorine group. But in fluoroquinolone, we have first and second generation. In second generation, we have gatifloxacin, sparfloxacin, levofloxacin. But in first generation, we have ciprofloxacin, which is very, very common, ofloxacin, and norfloxacin. These are commonly used fluoroquinolones, and their mechanism of action is blocking of the DNA topoisomerase 2, which is present in gram negative, and DNA topoisomerase 4, which is present in gram positive. Topoisomerase 2 is also called as DNA gyrase. What is the function of this DNA gyrase? Now, there is an extremely supercoiling between the two strands of the DNA. This positive supercoiling is converted into negative supercoiling, that is properly no other supercoils, via enzyme DNA gyrase. But fluoroquinolones inhibit these DNA gyrase only. So this positive supercoiling will not get converted into negative supercoiling. So this will persist. This will persist, and further there is difficulty in replication, transcription, translation, and so on, protein synthesis. So indirectly, it is a inhibitor. It is a replication antagonist. It prevents replication, and it is a topoisomerase inhibitor. Antiviral drugs, you have a various classification: herpes, influenza, and retroviral. In retroviral, you also have heart therapy, that is used for retroviral. In herpes, we have drugs like acyclovir, ganciclovir, that are basically analogs. They are resembling to certain analogs, like deoxyguanine analog is acyclovir is an analog of deoxyguanosine. Influenza, mainly two drugs, amantadine and rimantadine. Rimantadine is basically a methyl analog of amantadine. In heart, we have again four classification: protease, integrase inhibitors, integrase inhibitors, nucleoside reverse transcriptase inhibitors, and non-nucleoside reverse transcriptase inhibitors. Faultigravil is a most common example of integrase inhibitors. Protease inhibitors mainly include few drugs. It depends upon your syllabus. You don't have to study all the drugs. You just have to study few drugs that are inhibiting protease enzyme. Nucleoside reverse transcriptase inhibitors, you have zidovudine, lamivudine, didenosine. Non-nucleoside reverse transcriptase inhibitors, you have loviride and desloviride. They are called as non-nucleoside because they are non-competitive and they are reversible. They do not compete. They act on the allosteric side. Suppose if this is a site, and you can study it with the help of lock and key mechanism. So nucleoside reverse transcriptase inhibitors are similar in structure, but they prevent actual binding. Suppose if it's azidothiamidine like zidovudine, so it will compete with thymidine in triphosphate for binding on the active site. This is nucleoside reverse transcriptase inhibitors. But what will the non-nucleoside reverse transcriptase inhibitor do? It has an allosteric site here as well. So it will act on here and bring about the conformational change here. So now this won't be any furthermore able to bind on this site because of the conformational change done at the allosteric site. So it is a non-competitive. It is not competing with the substrate. Instead, it is binding at the allosteric site. That is antiviral drugs that you have to study. Next, we have anti-tubercular drugs. Their main example of anti-tubercular drugs are the price, you can remember it by the mnemonic price, that is pyrazinamide, rifampicin, isoniazid, streptomycin, and ethambutol. Other drugs are also used like capreomycin, kanamycin, para-aminosalicylic acid, rifabutin, ethionamide. But usually DOTS therapy is most common in in tuberculosis. So these are the few drugs that you have to study in tuberculosis. They have multiple mechanism of action, like some inhibit the mycolic acid synthesis, some inhibit the fatty acid synthesis, some inhibit the dihydropteroid synthesis, some inhibit the dihydrofolate reductase synthesis. So that depends upon the kind of drug that you're using. And rifampicin inhibits DNA dependent RNA polymerase. Rifampicin is the only drug from antitubercular that inhibits DNA dependent RNA polymerase. So you can remember the mechanism of action as well, and structures you have to study in medchem. You have no option, but in few drugs there are only structural highlights that would be mentioned in your syllabus. So you just have to study their highlights, and for sure those highlights may come in exam because they are very vital. Next is unit number 4, wherein there are three categories of drugs that are important, that is antifungal, anti-malarial, and sulfonamides. In antifungal, basically the most important classes are polyenes, allyl amines, and azoles. These are the most common categories that are used as antifungals, and the most commonly used are azoles. The azoles are further divided into two types, that is imidazole and triazole. The imidazole category basically consists of ketoconazole as an important drug, while the triazole consists of FIT-VIP, that is a acronym or a mnemonic for drugs. F stands for fluconazole, I stands for itraconazole, T stands for terconazole, V stands for variconazole, and P stands for posaconazole. In allyl amines, basically naphthene, terbinafine, these are important. Also, there is a special drug called 5-FC, that is 5-fluorocytosine, which is extensively the only anti-metabolite that is used in antifungal drugs. So this is a point that you have to remember, that 5-fluorocytosine is the only anti-metabolite used as an antifungal drug. In MCQ, it could be asked. In polyenes, the most widely used is amphotericin B. And amphotericin is highly lipophilic, and thus it is incorporated in the body via liposomes. That is why it is a liposomal injection, liposomal amphotericin B. Do not forget it. Other drugs are nystatin, hamacetin, but these are not that important. But amphotericin and nystatin are quite important. Antifungal agents are basically used to treat any fungal infection, any dermatophytosis, onychomycosis, tinea pedis, etc. So you have to remember each and every classification of the drug and the uses, which drug is used for which use. Next, we have anti-malarial. So in anti-malarial, there are basically different stages at which a drug can act, either at erythrocytic or at exoerythrocytic, or as sporocidal, or as gametocidal. In anti-malarial, you have different classification of drugs like 4-aminoquinolone, 8-aminoquinolines, quinoline methanol, biguanides, alcohol containing artemisia derivatives, quinine and the derivatives of quinine, diaminopyrimidine, and some antibiotics. So these are different drugs that can be used in malaria. Malaria is basically transmitted via female anopheles, and there are two vectors. One is the host and one is the a female mosquito. So you have to remember that there are two hosts. And you have to remember the life cycle as well, that how the malarial insect bites, how the merozoites gets converted into schizonts. Schizonts and trophozoites. These trophozoites are further, in the salivary glands, they migrate into liver, and then again they are multiplying. Then they are taken up by the another insect. They multiply, they form an ookinete, oocyst, and further cause infection. So you have to remember that the life cycle of the malarial parasite as well. So it is caused by the parasite responsible is a protozoa, that is Plasmodium vivax ovale falciparum. But Plasmodium falciparum is the is the most dangerous species that is causing malaria. So in 4-aminoquinolone we have chloroquine, hydroxychloroquine. In 8-aminoquinolones we have pamaquine, we have primaquine. In quinoline methanol we have mefloquine. In biguanides we have proguanil, which is a prodrug that gets converted into cycloguanine, which is the active metabolite. Alcohol containing most commonly used is halofantrine, lumefantrine. Artemisia is basically divided into two types: lipid soluble, water soluble. Lipid soluble is mainly artemether. These are basically some of the drugs that are developed in the CDRI, Lucknow, that is Central Drug Research Laboratory. And the water soluble is artemisinin, that was further converted into dihydroartemisinin. Quinine is a cinchona alkaloid. It is a levoisomer of cinchona plant. The dextroisomer is quinidine, that is used as an antiarrhythmic. So this is the most important question and the most confusing question that could be asked. Remember, levoisomer is anti-malarial and the levoisomer is quinine. In dextroisomer, in quinidine there is D, so dextroisomer is quinidine and it is antiarrhythmic. Please remember this cycle. In diaminopyrimidine, the most commonly used is pyrimethamine as a drug. So remember that. Now these drugs can be either used for clinical prophylaxis, suppressive prophylaxis, clinical cure, or radical cure, depending upon the stage in which they are used. But in some drugs, there are chances of relapse, that is reoccurrence of malaria. Also, do not forget an important ADR of this of the drug of few drugs, that is G6PD deficiency causing hemolytic anemia, that is the destroyal of RBCs. So in the people who are deficient of glucose 6 phosphate dehydrogenase, which is an important enzyme, G6PD, this is mostly in Africans and Negroes. It is quite deficient, so they often face the problem of hemolytic anemia. Sulfonamides are not that extensively used because of one fatal adverse drug reaction, that is Stevenson-Johnson syndrome. There are red patches, the swelling, the jaws, the neck. It is the most fatal adverse drug reaction, Stevenson-Johnson. Also, crystalluria can occur due to insolubility in water. Sulfonamides are classified as long-acting, short-acting, intermediate acting, and special purpose. In special purpose, basically you have mafenide, you have silver sulfur diazine, and sulfacetamide. Then you have sulfadoxime, sulfapyridine, sulfamethiazole, sulfamethoxazole, sulfa-isoxazole. So these are different sulfa drugs that are used, and their use has been quite restrictive. But yes, if a topical application for infections, dermal infections, they are still they can be still used, like mafenide. The basic moiety you cannot forget, that is a benzene ring attached with a sulfonamide group, that is the SO2NH2, and at para position another amino group. Similarly, you can also study the SCR. The amino group at para position, it should not be substituted, and it's substituted only in case of few drugs, but the activity varies, but mostly it's not substituted. However, the amino group that is attached with sulfur, the one hydrogen can be replaced with a heterocyclic ring, that can be pyridine, that can be pyrimidine, that can be isoxazole, then can be methoxazole, anything. So just remember, this is the basic moiety of sulfonamides, and that's all from unit 4. Next we have unit 5. In this, basically you have a little higher and a complex level of study, that is the docking, molecular docking, pharmacophore modeling, protein remodeling, and the drug designing. So whenever we think of designing a drug, there are various physicochemical parameters and certain few different evaluation parameters that are kept in mind in accordance with designing the drug. For example, if we have the Hammett's electronic parameter or Hans analysis. So in Hans analysis, there are various steric factors involved. There is TAF's analysis as well. The electronic, steric, and lipophilic. These are the three basic parameters that are useful in drug designing: electronic, steric, parametric. So you do not have to forget these three parameters. Docking. Whenever we do docking, first we have to find a hit. What is a hit? It is a ligand or a drug, we can say, that sorry, there is hit identification and a lead optimization. So hit is basically a target of the ligand, a target of the drug, where we have to give that drug. If it's anti-malarial, so if what is the mode of action, what is exactly the site of action, the target of action. Then is the lead optimization. We have to find a lead molecule, a drug or a ligand that can actually bind on that target site. And then drug finding, and various parameters are considered: the lipophilicity, solubility, steric factors, electronic factors, parametric factors, lipophilic, Lipinski's rule of 5. It is very important whenever we consider a drug. The log P should be less than 5 for a water soluble drug. Along with that, the hydrogen bond donor should be less than 5. The hydrogen bond acceptor should be less than 10. So you have to consider all of these parameters whenever you're designing a drug. So that's all for today's video. Thank you so much for watching the video.